Welcome back to BioMindscape. Today we are going to solve some PYQs. So here the CUET questions are there. So which one of the following disease can be corrected by the gene therapy? So here the answer is cystic fibrosis, not sleeping sickness or measles or AIDS. Or it is a classic example of monogenic inheritance disease. It can be caused by the mutations in CFTR genes, and which leads to the production of faulty protein channels, and the gene therapy that aims to deliver a correct copy of CFTR genes to the cell of the patient. Particularly, therapy aims to deliver a correct copy of CFTR genes to the cells of the patient, particularly in the lungs, to restore the normal function. So it is a primary candidate for correcting the gene therapy. So this is the reason why this answer is correct. Now moving on to the next question. Here we can see the brain and spinal cord develop from the ectodermal arrangement in a tubular structure. They are called the neural tubule. The anterior part of the neural tubule that expands and construction appears that create multiple regions, arrange the parts of the embryonic brain chamber in 5 weeks of embryo from top to bottom. So here first is which one? E, that is Telencephalon, E, then Diencephalon, then Mesencephalon, then C is the Metencephalon, and B, that is Myelencephalon. So the development of the central nervous system begins with the formation of the neural tube, the anterior. First, the anterior, what happens here? Anterior end of the tube that undergo different forms of brain, and the 3 primary brain vesicles they are formed, that is a forebrain, midbrain and hindbrain. So by the 5th week of the development, these 3 vessels that fully develop, and hindbrain by the 5th week of development, the 3 vessels that further differentiate into the 5 secondary vessels. And we have to tell the correct order from top to bottom. So first what happens? First is the Telencephalon, T, from top to bottom. Telencephalon, then Diencephalon, TD, then D is the Mesencephalon, then C is the Metencephalon, and Myelencephalon, that is B. So last option is Myelencephalon, that is the last one. Next question. Southern blotting process does not include what? Digestion and separation of DNA fragment using the agarose gel, electrophoresis, ligation of the targeted DNA into the cloning vector, transfer of DNA fragment from the nitrocellulose membrane. So here the correct answer, Southern blotting produces that does not include the hybridization of the membrane with the label probe. So here ligation of the targeted DNA in the cloning vector, that is the correct answer, that is not included in Southern blotting. Whereas the digestion and separation of DNA fragment using the agarose gel, that is used in the Southern blotting. Then transfer of DNA fragment to the nitrocellulose membrane, that is also used, and hybridization of the membrane with label probe. Because I know A, C, D, they are the process in the Southern blotting. So the B, that is ligation of the targeted DNA in the cloning vector, that is not the correct one. We know that steps: first digestion and separation, then blotting happens, then hybridization of the probe happens, and then detection. Here we have to tell which one of the correct sequence of the steps in the genetic engineering of the E. coli for insulin production. So here the 4 options are: obtain a copy of human insulin gene by isolating the mRNA, switching on the gene action, identifying the transformed bacteria prior to cloning, then inserting the DNA into the plasmid vector and inserting the plasmid vector into the host bacterium. So here we have to choose the appropriate option. So here the option is First is obtaining a copy of human insulin gene by first we will isolate the mRNA, obtain a copy of human insulin gene. Then the second option is we will insert the DNA into the plasmid vector, and inserting the plasmid vector into the host bacterium, that plasmid vector will be inserted into the host bacterium, and after that it will be identifying the transformed bacteria prior to the cloning. Prior to the cloning we will identify the transformed bacteria, and after that the switching on the gene action is the last step here in the E. coli. These are the steps of E. coli for insulin production. So A, D, C, B is the correct answer. A multiple cloning site. What is a multiple cloning site? It allows multiple choices for choosing the restriction enzyme for the cloning. It allows multiple choices for choosing the restriction enzyme for cloning. First option is allow contain several copies of cloning gene. Allow multiple choice for choosing the organism for cloning. Then contain multiple copies of the same restriction enzyme. First is MCS. It is containing recognition sequence for number of different restriction enzyme clustering together in one location. Why A is incorrect? So first option, what is it? Contains several copies of cloning gene. What does a multiple cloning site contain? First option is incorrect because MCS, it is a site where the gene is cloned and it doesn't contain the gene initiation. Now why B is incorrect? The choice of the organism, it is determined by the other things other than MCS. C is correct. Why? It is a primary function of MCS and to offer the selection of unique restriction sites. Whereas D is incorrect. Why? MCS contain the sites for many different enzymes, not multiple cloning. What is MCS? Multiple cloning site is MCS. It is a single site for cloning that offer multiple options. What does this site do? They recognize the sequence. Next question is, which one of the following best describe the application of DNA microarray? So here the answer is first one, second one, third one or fourth one we have to tell. The first option is to investigate the level of gene expression in a cancerous cell. This is correct because it describes the application of DNA microarray. It helps in investigating the level of gene expression in a cancerous cell. DNA microarray is a powerful molecular biology tool used for highly throughput analysis, and it consists of the solid surface like the glass slide onto which thousands of different sequence probes are spotted in order of the array. So here 8th option is correct. Next question is, what are chemokines? What are chemokines? Option A, cell surface receptor. Option B, specialized chemotactic cytokine. Option C, any foreign antigen. And option D, chemical base antibodies. So here the chemokine is a specialized chemotatic cytokine. That is B option. Cytokines are a broad category of protein that are that can be a chemical messenger. That are the chemical messengers between the cells and the immune system. Or this chemokine, they are specific subfamily of cytokine. The primary function is to induce the direction cell migration process, and we call it as chemotaxis. Immune cell like neutrophils, macrophages, they follow the concentration gradient of the chemokines to move from the blood to the tissues. It moves from the bloods to the tissues and towards a site of infection or inflammation. Therefore, a chemokine is specialized cytokine that has chemotactic cell attracting activity.
Thanks for watching. Do like, share and subscribe BioMindscape.